Technical Veterinary Learning TVL channel. The founder and the producer is the Prof.Ahmed Mamadouh El-Sherif, Professor of Infectious Diseases and Epidemiology, the ex-head of Department of Veterinary Medicine, the ex-vice dean of Faculty of Veterinary Medicine, Bani Suef University, Egypt. We will speak today about the infectious disease process with complete discussion and examples about this subject. اسم الموضوع باللغة العربية هو عملية حدوث المرض المعدي. هذا ما ستناقشه هذه المحاضرة بالتفصيل مع بعض الأمثلة الضرورية. The other name of this subject is the chain of infectious. This synonym will be found in several of textbooks, but I prefer the first synonym, the infectious disease process, as it describes briefly and completely the process of occurrence of any infectious disease either in human or animal science in complete manner. الاسم الآخر لهذا الموضوع باللغة العربية هو سلسلة العدوى، ولكني أفضل الاسم الأول وهي عملية حدوث المرض المعدي، لأنه اسم مختصر ولكنه كامل التوصيف لأي مرض معدي سواء في الإنسان أو الحيوان. Infectious disease process occurs when the pathogenic risk factor or the pathogen or the microbial agent leaves its reservoir or host, either specific or non-specific, through different portal of exits that differentiated according to the pathogen and the disease, and by some mode of transmissions, either direct transmission or indirect transmission or by contagiousness. They enter through an appropriate portal of entry within the reservoir specific or the host specific animal or human to infect them. This sequence or this process is named as infectious disease process. This process occurs with any pathogen that cause a disease in human being or in animal species all over the world and through the history. This a symbolic picture of infectious disease process. Explain that number one in this process is the pathogenic risk factor or the microbial agent or the pathogen. And number two is the reservoir or the host, either specific or non-specific. And the third part is the portal of exit. And then the fourth part is the mode of transmission. 5 steps on the way is the portal of entry, and the 6th way is the susceptible reservoir or the host, where the agent finally reaches to it again to cause the disease. The infectious agents or pathogenic risk factors, or in other words, the essential cause factors. Bacteria, viruses, fungi, parasites, prions, rickettsia. The pathogenic type of them is the direct pathogenic risk factors, but in some cases during the immunity of mammals, as human being or animal species is dropped under any stress factors, some of the commensals of them is changing from non-pathogenic nature into pathogenic nature. And because of this, we call them pathogenic risk factors. Some textbooks, including the poisonous creatures as an infectious agent. Important factors linked to any infectious agent or pathogenic risk factors are adaptability, host range, virulence, pathogenicity, and ID50 and LD50 or the dose. The adaptability including the adaptability of the pathogen on a specific tissue, by means that how this pathogenic factor like Mycobacterium tuberculosis or Mycobacterium bovis or Mycobacterium ovis adaptable to different tissues of body organs to cause the infection. The host range here is a difficult task, as when the pathogenic risk factor has a wide host range, by means that it can infect more species in the same time, this makes it most dangerous in its pathogenicity. The virulence of the pathogenic risk factors detect how they can produce the disease signs or symptoms. Virulence of Bacillus anthracis is more than those of dermatophytes in its production for a severe clinical disease reached by animal or human to the death. This virulence will depend upon the immunity of the host or the final host, as well as depend on the pathogenic factor itself. Pathogenicity and how the microbial agent can produce its effect inside the cell, how to replicate themselves inside the infected cells, how to enter the different organs, and how many times they can multiplicated inside the cells as Procella microorganism who when enter through the skin, one cell can be reached to hundreds within hours. The dose of the infection as ID50 or LD50 detect and determine the dangers of the pathogen. When one pathogen can produce the disease, it is more different than thousands of pathogens were needed to cause the infection. The first example is more dangerous than the second one. The reservoir of an infectious agent is the habitat where these pathogenic agents normally live, grow, and multiply. Reservoirs includes animals, humans, and environments. These reservoirs may or may be not the source where these infectious agents are transferred to its final host. For example, Clostridium novii type D, the main reservoir is the soil, as in all clostridial microorganisms. But the source of it is the cereal grains that given to a sheep during flushing. In presence of some factors that it changed the environment inside the rumen from aerobic one to anaerobic one, where multiplication of these type of clostridial species become high and cause toxin production in huge amount and ended by struck disease with subacute signs of deaths of several of these sheep within hours. Reservoir may be responsible for or not responsible for transfer of an infectious agent to a host. As example, grasses on the sides of water tunnels in enzootic areas, such as in Egypt, India, Sudan, and other countries that report or has reservoir may or may be not are responsible for transfer of an infectious agent to a host. As examples, grasses on the sides of water tunnels in enzootic countries with fasciolaces, such as Egypt, India, Sudan, and other countries. Are they responsible for transferring of the encysted metacercaria of fasciola to grazing buffaloes or cattle as well as sheep? Soil is an example of main reservoir to the Clostridium botulinum species, as the soil here acts as the improperly foodstuff contain the different agents of Clostridium and it contaminated by soil, and the most clear example here is the Clostridium botulinum, where in contaminated food stuff that contain Clostridium botulinum spores with presence of anaerobic conditions, these Clostridium botulinum will multiplicate and producing the toxins that cause toxicity of the herds feed on these contaminated stuff as sheep or cattle. Types of reservoir: number 1, human reservoir; number 2, animal reservoir; number 3, environmental reservoir. Human reservoirs. Most common infectious diseases have human reservoirs. Diseases that transmitted from person to person without any type of intermediates, including sexually transmitted diseases, measles, mumps, many respiratory pathogens as Mycobacterium tuberculosis, streptococcal infection, smallpox, as human were the only reservoir of these smallpox virus. Human reservoirs are two types. Number 1, symptomatic persons. Number 2, carriers. Symptomatic persons, they are aware of their illness. They have low rate of transmitting infections. They are either too ill to become out of their homes, and they are taking precautions to reduce transmission or receive prescribed treatment which limit the disease. A carrier is a person with an apparent infection, so he can transmit microbial pathogen to others as he don't realize the infection and consequently take non-special precautions to prevent transmission of infection. Classification of carriers. Number 1, asymptomatic or passive. They are healthy carriers. Number 2, incubatory carriers. Number C, convalescent carriers. Number 4, chronic carriers. Asymptomatic or passive or healthy carriers. Those persons never express symptoms despite being infected. Here is a picture taken in Veterinary College of Pennsylvania University at October 2019 during a festival about October War, where all the students are gathering each other in this collection. Maybe one of them have flu, but in incubation period without appearance of clinical symptoms. Maybe later some of them showing flu signs. Incubatory carriers. They are persons that can transmit any pathogenic agent during the incubation period before clinical illness begins. This is a picture of animals all over the world during the corona pandemic, where there are an employee taking the temperature of all entrance persons. Any person of them showing fever or low grade fever is taken directly to the hospital and prevented from coming together to other peoples. Convalescent carriers. Those are persons who already recovered from new illness but remain capable of transmitting their infections to others, as salmonellosis, typhoid and or paratyphoid disease, which could be transmitted from person to person through shake hands. Chronic carriers. Those persons continue to harbor a pathogen such as tuberculosis, hepatitis B virus, or Salmonella typhi for months or even years after their initial infection and continuing for their transmission to others. Animal reservoirs. There are two types of animal reservoirs. Number one, symptomatic animals. Number two, carriers. Symptomatic animals. These animals, their owners are aware about their illness, so they are likely lesser to transmit their infection. Their owners are isolate them to contact to others and take precautions for reducing transmission and or receive treatment which decrease their diseases. Animal carriers are animals with an inapparent infection, so they can transmit pathogenic risk factors to other animals. Their owners don't realize that they are infected, so consequently have no special precautions for prevention transmission of the diseases. The ideal here example is the MERS-CoV virus that appeared in Saudi Arabia as endemic disease for camels at 2012, where it distributed through all collections of camels there, as well as it acts as a zoonotic disease which transmit the respiratory form to contact persons. Classification of animal carriers. There are 5 types. Number 1, asymptomatic or the passive or healthy carriers. Number 2, incubatory carriers. Number C, convalescent carriers. Number 4, chronic carriers. And number 5, zoonotic carriers. We will speak about each one of them laterally. Asymptomatic or passive or healthy animal carriers. These animals never express symptoms despite being infected. The most clear example is the BVD. Incubatory animal carriers. They are animals who can transmit the pathogenic risk factor during the incubation period before clinical illness begins. The ideal example here is the 3-day sickness between large ruminants. Convalescent animal carriers are those animals who have recovered from their illness but remain capable of transmitting the pathogenic risk factors to others either directly or indirectly. The ideal examples here are cases of babesiosis and theileriosis. Chronic carriers are the animals who continue to harbor a pathogenic risk factor such as tuberculosis, Escherichia coli, or Salmonella typhi for months or even years post their initial infection. Zoonotic carriers. They are animals with an infectious disease that is transmissible under natural conditions from vertebral animals to humans. The most clear is the rabies. Also, there are newly emergent zoonotic diseases in North of America, including West Nile encephalitis, and between some collections of peoples, the monkeypox. Many newly recognized infectious diseases in humans, including HIV or AIDS, Ebola infection, and SARS, are thought to have emerged from animal hosts, although those hosts have not yet been identified completely until now. Zoonotic diseases such as brucellosis, anthrax, plague, trichinosis, tularemia, rabies, West Nile encephalitis, monkeypox, HIV or AIDS disease, Ebola, SARS, and tuberculosis. Zoonotic diseases are about 273 diseases that can be transmitted from animals to humans. Environmental reservoirs are soil, plants, and water. Soil reservoir for histoplasmosis, anthrax, internal parasites, eggs of external parasites, Clostridium diseases, and etc., where all these pathogenic risk factors live and multiply in the soil. Plant reservoirs, where brucellosis, tuberculosis, E. coli, Clostridium botulinum, and etc. live and are transmitted by infected plants. In case of soil or plants or water reservoirs, the geographical area from part to another throughout the world are different, by means that histoplasmosis present in some American cities, internal parasites present all over the world, brucellosis present in endemic areas, tuberculosis more in Africa, and now in Europe. WHO at 2022 elaborated that it is the coming pandemic through Europe countries, and so on. Water reservoirs, leptospirosis, foot and mouth disease virus, PPR viruses, glanders, and etc. are pathogenic, live and transmitted in the water from infected animal to another one. Portal of exit. The portal of exit means the path by which a microbial pathogen leaves their hosts. The portal of exits usually corresponding to the site where the pathogen risk factor is localized. For examples, influenza viruses, Mycobacterium tuberculosis, infectious proven rhinotracheotis virus exit the respiratory tract. Schistosomiasis exit through urine. Leptospirosis exit through urine. Sarcoptes species exit through skin lesions, either in human or animal species. Rabies virus exit through saliva. Some blood-borne agents can exit by crossing the placenta from mother to fetus, such as toxoplasmosis, syphilis, brucellosis, while others exit through cuts or needles in the skin. Hepatitis, blood-sucking arthropods such as babidiosis, filariasis, and malaria. Mode of transmission. Any infectious microbial pathogen may be transmitted from its natural reservoir to its susceptible host by different ways. These different ways are calling the transmission way. There are two ways. Number one, the direct transmission way. Number two, indirect transmission way. Direct transmission way mean that an infectious pathogen or risk factor is transferred from an available reservoir to a susceptible host through direct contact or by droplet spreading. Direct contact occurs through skin to skin, licking, bites, sexual intercourse, transplacental transmission, flesh eating, contaminated soil, vegetation harboring infectious pathogen such as mange, rabies, bovine viral diarrhea, cholestridiosis, colibacillosis, etc., etc. Droplet spreading refers to spray with relatively large short-range aerosols produced by sneezing, coughing, which reach to 10 meters long, such as coronavirus, IBR virus, mycotuberculosis bacteria. Indirect way of transmission refers to transferring of an infectious pathogen from a reservoir to host by one of the followings. Number one, suspended air particles. Number two, inanimate objects or vehicles. Number three, animate intermediaries or vectors. When we speak about suspended air particles, this means airborne transmission occurs when infectious agents are carried by airborne dust or droplet nuclei suspended in the air. Airborne dust includes materials that have settled on the surface and they become resuspended by air currents, such as Mycobacterium tuberculosis, sporotrichosis, and etc. Infectious particles blown from the soil by the wind, such as Ascaris egg, Trichoviricosum, fungal agents, and etc. Suspended air particles refers to droplet nuclei are dried residue of less than 5 microns in size. Droplet nuclei may remain suspended in the air for long periods of time and may be blown over great distances, such as foot and mouth disease viruses. Inanimate objects or vehicles denoted to indirectly transmit an infectious pathogen, including foodstuff, water, biological products such as blood and or serum, and so on, as in case of foot and mouth disease, glanders, and brucellosis. Number 2, vehicles that may be indirectly transmit an infectious pathogen, including vomits. Inanimate objects such as blankets, beddings, and or surgical scalpels, as in case of ringworm, strangles, epizootic lymphangitis, and etc. Number 3, vehicles may provide an environment in which the pathogen grows, multiplies, and or produces toxin, as improperly contaminated foods provide an environment that supports production of botulinum toxin that caused by Clostridium botulinum, which leads to botulism disease in ruminants. Inanimate objects or vehicles denoted to vectors such as mosquitoes, fleas, and ticks that may carry a pathogen through purely mechanical means or may support growth or changes in them. Number 2, vectors can pass the pathogen to the next generation transovarially. Transovarian transmission of filariasis, babesiosis, and anaplasmosis. Number 3, vectors that have metaforces can pass pathogen from a stage to another. Transtadial transmission, as it is without any changes, as in plague, swine plague, and so on. Number 3, animate intermediases or vectors. Vectors are invertebral spaces that transmit infectious microbial pathogen actively between different hosts. Vectors such as mosquitoes, fleas, ticks, tapenos may carry the pathogenic risk factors through purely mechanical means or may support growth and multiplication or support changes occur in them, such as babesiosis, blue tongue disease, Rift Valley fever, and etc. The way number 5 in the process of infectious disease occurrence is the portal of entry. The portal of entry refers to the way of a pathogenic risk factors enters to susceptible host or hosts. It must provide access to tissues in which this pathogen can multiply or producing toxin. Usually, these microbial pathogens use some portal to enter a new host that they use to exit the host of the source. Examples of that: influenza virus, Mycobacterium tuberculosis exit respiratory tract, Brucella organisms through vaginal and uterine discharges, foot and mouth disease virus through saliva, schistosomiasis through the urine, cholera vibrio through fecal matter, Sarcoptes species either in human or animal through skin lesions. The sixth step in the infectious disease process is the host. Susceptibility of hosts depends upon: number one, genetic makeup of them. Number two, their specific immunity. Number three, non-specific factors that affect an individual's ability to resist infection or to limit pathogenicity. The genetic makeup may be increase or decrease the susceptibility for certain pathogens. As an example, native breeds of cattle, buffaloes in Egypt, showing highly resistant to different infectious diseases than imported ruminants. Number two, people with sickle cell traits seem to be at least partially protected from particular type of malaria in case of humans. The specific immunity of hosts refers to the protective level of antibodies that are directed against certain or specific pathogens, such as antibodies may develop in response to infection. Number two, antibodies developed due to vaccination. Number three, toxoid, by means toxins that has been deactivated but retains its capacity to stimulate production of toxin antibodies. The non-specific factors for the hosts. Non-specific immune responses, general action of white species against injury, stabbing with foreign bodies, abscess reaction. Number two, the intact skin and mucous membrane act as a barrier against pathogen infection from outside. Number three, gastric acidity, kill most of pathogens that enter through mouth. Cilia in the respiratory tract and the cough reflex act as non-specific factors that decrease the pathogen stability on the tissues of the trachea, bronchi, oropharyngeal area, and or lung tissues. Factors that may increase susceptibility to infection by disrupting host defenses. Number one, malnutrition in animal and human. Number two, alcoholism in case of human. Number three, disease impair the non-specific immune response, such as BVD virus in animals and HIV virus infection in human. Number four, therapy that impairs the non-specific immune response, such as high doses and repeated of dexamethasone injection for long periods of time. In the following, I will give you examples for infectious disease process. In case of foot and mouth disease virus, natural host or asymptomatic is the pigs. Transmission hosts moderate to severe symptoms are the small ruminants, sheep and goats. Spillover hosts severe infection with high mortality are large ruminant of cattle and sometimes buffaloes. MERS virus. Natural host or asymptomatic host is the pet. The transmission hosts moderate to severe symptoms will appear on it are camels. Spillover host where severe infection and high mortality rate has been noted, it is the human beings. Hendra virus. The natural host and asymptomatic appearance of any signs are pets. The transmission host where moderate to severe symptoms appear are the equines. Spillover host where severe infection and high mortality appear on them are human beings. In case of Mycobacterium tuberculosis, the natural hosts where asymptomatic appearance of the disease are human beings and all warm-blooded animal species. Transmission hosts where moderate to severe symptoms appear on them also are human beings and all warm-blooded animal species. Spillover hosts where severe infection and high mortality rates are noted are human beings and all warm-blooded animals. So Mycobacterium tuberculosis is a dangerous. Infectious disease process of babesiosis. The pathogenic risk factors are Babesia species. The reservoirs are ticks and all animal species. Portal of exit is the blood, transovarian of ticks. Mode of transmission is indirectly through blood-sucking arthropods or surgical instruments and or needles. Portal of entry are blood vessels. Hosts are ticks and all animal species. Infectious disease process of colibacillosis. Pathogenic risk factor is the Escherichia coli. Reservoirs are all animal species. Portal of exit is the fecal matter and mastitic milk. Mode of transmission are direct and indirect contact. Portal of entry are mouth, other orifices, and or umbilicus. Final host are all animal species. This is applied exercise. Information about monkeypox disease provided on the following pages. After studying these informations, outline the chain of infection by identifying the infectious disease process as understanding from the following article that published by NBC News channel with the following link: https://www.nbcnews.com. Please keep on the track with me for the most modern informations about different infectious diseases and epidemiology from subscribing this channel and following up our videos. Thank you and see you later, alligators.